I wouldn't make you come to class for 2 slides. If it were 2 slides, I'd make that little 5-minute video and send it out to y'all and be done. Right, but we got more than that. Okay. Come on.
I didn't even know how to pack a water. Yeah, you took a well, I have a water.
Oh no. The computer just froze.
Perfect.
Oh, it's back. It's back. Okay. So.
I know.
Good morning. You have to give a best man speech for a class?
No, I have a wedding.
Oh, you actually have, okay. Okay, I was so confused.
I'm such a weenie that I waited to take it until my last semester.
Oh, well, that's okay.
She was hoping they were just like, you don't have to.
Yeah, I was hoping they were like, yeah, it's okay.
You put it off till the end.
And
Well,
for freshman studies, they make you.
oh, really?
Hey,
Here's the library.
So like, it wasn't.
Yeah, but you got it covered, right?
I think that's after enough Mark Love presentation.
Mark Love. Who's
Oh,
Mark
yeah,
Love?
the library guy. He's the most famous guy in the
The biology.
You've never heard of him?
I've never heard of him.
We should have told her she's in schedule.
I'm
Like,
not a
like
problem.
all the biology teachers like will schedule him to like come and do like
A databases tutorial,
Yeah.
basically.
Oh,
like
and how many times have you done it?
like
So many times. He's like the go-to person to schedule with. Immunology literally had a big.
Yeah, and that's like a 400 level class.
So-
like, if you don't know how to use
So from 200 to 400. It's my
Oh,
love.
every single
200.
sometimes you have like a whole week of Mark Love, like every
Oh my God, that's horrible.
Oh, yeah.
That's horrible. No.
It's like a Where's Waldo competition, except Waldo is right in front of your face.
You don't have to really find him, right?
Yeah, no, he's just there. He shows up. He's like,
Not even wearing
he's
his
Nicholas'
getup.
local cryptid, basically.
I think that's like the universal skip day. Mark Love just comes out, present, you just skip the class.
You're like, not coming.
Same thing every time. Yeah, he's like, hey guys, and I'm like, hey again. I'm like, as if you don't see my face. I'm not wearing green because I actually green, but I have a green highlighter.
There you go.
So I'm
There
in
you
the
go.
spirit. Yes. And I have green lip stickers. I
Okay.
don't have a green shirt.
And I need chemistry. I stand here chatting and then I don't prep our stuff. Okay. We were like at the very end, right?
We had like 3 slides left.
Yeah.
If you have a question about that,
Yeah. And how they work?
kind of, yes.
Okay, so let's do the last 3 slides and then we'll walk through how statins work. And then if you have a question, ask me at that time.
Speaking of
Yeah, yeah.
stat, yes, I did look
Absolutely.
further into it. There is a stat there. prevention of COVID.
There is. Okay, send it to me. I want to see.
Well, I'm overdue for one.
I have to.
Really?
Really?
Yeah, some people can, some people can't. It's okay. What am I doing? So I have to mirror this. I'm the wrong thing. Here we go. Here we go. So check in on iClicker.
Are we going over the new chapter or we're just over new stuff?
Where's? No, we're still on 16.
I know, but I want to. Yeah, for rest of class.
We're 60.
16. The
60.
whole class.
And then the exam's the next day.
I want to
Yeah,
make
yeah,
sure
yeah.
because I didn't print out the slide 17.
No. We're not starting 17 until after the exam because that's
Oh my
just,
goodness.
yeah, no. We might end up
You're like
17
who's done that.
17 is like 4 days. Like, well, it's 3 full days and a half day. We might try to make it less so that we have more time on 19, but we'll see. But no, I don't want to go on to the next one yet.
17 and 18 for the next plan, and then 19 is part of the fourth plan.
Yes,
Or
19
final.
19 is the accumulation of all of the metabolism that we have studied.
Gotcha.
So it is essentially combining everything and reviewing everything. And so it's like perfect because it like is the final.
So, just the like compilation.
Yeah. Yeah. Okay.
What time?
Well,
Somebody's picking their iPhone.
oh, okay. Okay, we'll talk about it after.
We get lost sometimes.
We'll talk about it after. All right. So, right, that was homeostasis. We did that one, right?
Yes.
We finished that
On
one.
the
Okay.
next one.
All right. So, remember when we talked about what's the rate limiting step in cholesterol metabolism? CoA reductase. Okay, so if we look at the substrate for HMG CoA reductase, this is HMG CoA, right? This is the natural substrate right here. This is the natural substrate for HMG CoA reductase. Okay, all of these cholesterol lowering drugs, look at the red portion of each molecule. Maybe it'll be easier to see if I turn off the lights in the dark. I know I always do this wrong. Oh, and I didn't say I'll do it.
Oh, yeah.
Why couldn't the elephant use the computer? He was afraid of the mouse. He was afraid of the mouse.
True. Like, I know it's like in cartoons that elephants are afraid of mice, but is that actually true?
I don't think they're afraid of them.
like
I don't know.
Maybe you should go find them and
I don't know.
see.
Yeah, I don't know.
I was just
Don't know. not in my wheelhouse.
I'm a monkey's ass, I guess.
Right, exactly. So when we look at these different substrates, right? All of these different ones, some of them were isolated from fungi, some of them were chemically synthesized, right? And remember, we've talked about in the past, you know, like with antibiotics, Mother Nature has so much more time on us, right? So a lot of the drugs that we use have actual natural origins, basically, and usually we just chemically modify them, right? So what you can see is that the substrates are very similar to HMG CoA, so they can act as inhibitors. So the rest are inhibitors of the HMG CoA reductase. But that's not the only thing that these things do. So if we think about this in the long term, all the implications of inhibiting HMG CoA reductase. And before we can list all of them, which I want to do at the end, we also have to talk about how we regulate all the different components of cholesterol synthesis and metabolism. So we're going to talk about that, and then we're going to come back to these drugs and what their overall effect really is. So if you think about, in general, flux, let's talk about this. Flux through lipid metabolism. OLIS, metabolism, right? So think not just about cholesterol, think about all lipids. There are 3 big major cellular things that we have to worry about in whether or not we're going to synthesize lipids, we're going to break them down, we need to make cholesterol, we need to make all the different things. So the number one, when we think of why would we make or break down fats, what do you automatically think of first? Energy charge. Oh, y'all are right on the wavelength. I'm loving it today. All right, so energy need. You need, right, energy charge of the cell, right? Okay, now I want you to think about other reasons why we need lipids. And when I say lipids, I mean all lipids, not just triacylglyceride. Cell membrane. So what if you have something that's like a really rapidly dividing cell? You're going to need a lot of membranes. So membrane requirement for, we'll say, rapidly dividing cells. Okay, so we did like triacylglycerides, we did phospholipids, sphingolipids, right? Now what's the third? So I heard a couple of things. Endocrine signaling. Why, like, what kind of lipid is going to be required for endocrine signaling? Steroids, eicosanoids, they're all derivatives of cholesterol. So what are the others? So we're going to say need for cholesterol derivatives. When we say cholesterol derivatives, I'm going to say in general steroids, right? That would encompass everything that y'all have said so far. But there's something else also that is a bile acids. So bile acids, sure. So these are the things that the cell has to be sensitive to turn things on or off, right? Okay, so the question is, do you want to have all the enzymes that are required to do all these things present and ready to go in the cell all the time? No, that would be a huge energy drain for the cell, right? So the cell's always trying to think about energy optimization. So how could we create a system where we just have the genes when we need them? Transcription factors, transcriptional control. So that's what we're going to talk about here, is now transcriptional control. So yep.
So does steroids also encompass the eicosanoids?
That's kind of the big general. They all come from a common derivative. So that we don't have to name each individual thing. If we just say it's a cholesterol derivative, it kind of covers everybody. All right, so transcriptional control, as I'm hiding it. So transcriptional control is what we want to be able to use to regulate the enzyme levels, right? But now, do I want a bunch of different transcription factors? No, it would kind of get messy. I want to have one transcription factor or a couple of transcription factors that will regulate multiple enzymes within the same pathway. We talked about this in prokaryotic systems in Biochem 1. Do you remember this? Vaguely? Operons? Do you remember operons? Right? Where you had one transcription factor and then you had like 3 genes in sequence, and the one transcription factor made all the genes. Yeah, so it was 1 promoter and then 3 genes. So this is a little bit different. It's not quite like that. It's really 1 transcription factor, but do you just make 1 transcription factor molecule? No, you're going to make a ton of them, right? And then that same transcription factor is going to go to multiple genes. So each gene has its own promoter, but the promoters have homologous sequences. Does that make sense? I feel like I lost y'all a little bit. No? Yes? Good?
1 per minute.
1 transcription factor that recognizes multiple promoters, but all of those promoters regulate the expression of genes that are involved in lipid metabolism. Sort of, yeah. Yeah, okay. So this is called SREBP. What is SREBP? It's sterol regulatory element binding proteins. So these are basically transcription factors. So normally they're embedded in the ER membrane, but if you decrease the amount of cholesterol in the cell, it is going to promote the binding of these molecules to specific sequences, promoter sequences, to induce transcription of genes that are involved in metabolism of lipids. So in this case, the sterol regulatory element binding proteins, right? We're looking at the LDL receptor gene, right? So if I have low levels of cholesterol, do I want to have more LDL receptor? So you can take it out of the blood and extract out the cholesterol, the lipids, the whatever it is you need in there. So yes, we do want to upregulate LDL receptor genes. Do we want to upregulate HMG-CoA synthase and HMG-CoA reductase? Yes, because this is de novo synthesis. So if you look at these, right, these two genes are de novo. And then what would LDL receptor gene be? Dietary extraction, right? So you're going to either increase, you're going to do both. You're going to increase dietary extraction and you're going to increase de novo synthesis. New.
From scratch.
Yeah, from scratch. It just means
Oh,
from scratch.
it's
It's Latin. I believe it's Latin. Okay, so if we increase the LDL receptor, right, we increase the receptor protein on the surface. The more receptor you have on the surface, the more you're going to endocytose those LDL particles, right? And you remember we were talking about really briefly that there are some people who had a genetic disorder where they expressed more of this LDL receptor, like 5 times the normal amount of this receptor. And what happened to those people? Heart attacks at like 5 or 6 years old. Crazy. But you do need an ability to regulate this normally in a regular cell, right? But if you abnormally increase it, it's bad.
And with that, because you had so many on the surface that
It's-
wasn't actually functional.
So you had so many on the surface that you're pulling so much in that now you just have too much. You have too much cholesterol. Mhm. Okay, so then HMG-CoA synthase and HMG-CoA reductase, right? These are the enzymes that are de novo synthesis. So we have increased flux through the cholesterol biosynthetic pathway, right? That's just talking about de novo synthesis. But in combination, both of these two things are going to increase our intracellular levels of cholesterol. And that's what we wanted, because what initiated this? Low levels, right? But do decrease the level of serum cholesterol. Yes, yes. Right. So if this happens, right? If this happens normally, but then you're taking a statin drug, which side of this pathway is inhibited? This is inhibited, right? And so if this is inhibited, and which one are you going to get more cholesterol out of per day? Synthesis, you get like a gram, and here you get like 0.3. So if we inhibit this side, now you're going to pull more LDL molecules out of circulation, and you're not going to make it. So overall, you're going to have a lower level of cholesterol in the body, and you're going to have less floating cholesterol, basically. Yeah. Okay. So how does this work? All right. So do you remember in Biochem 1, we talked about certain enzymes starting out as precursors, and then those precursors have to be activated in some way, shape, or form. And proteolytic cleavage was one way that that happened. This is similar to that, except it's more complicated, of course, right? So you have this here, right here is our transcription factor. Just this little, it looks like an upside down heart, right? Just that little part is the transcription factor. So this entire thing is embedded in the ER membrane, and it's bound to an inhibitory complex called S-cat. S-cat like holds on to it and prevents it from going anywhere. And then N-sig binds to S-cat. And so all three together, if you have all three together, nothing happens. They stay in the ER, okay? But the only way that N-sig and S-cat stay is if they're bound to cholesterol. So at high levels of cholesterol, this is what you have, right? So it binds S-cat. It's just, you don't have to know like the abbreviations for all the, you don't have to know all that. But what I'm really worried about is this one right here, okay? So these just basically hold our transcription factor in the ER membrane so that it doesn't go anywhere if you're bound to cholesterol. Now, what happens when serum cholesterol levels get low? The intracellular cholesterol levels get low, right? So then what happens is N-sig is going to disassociate from the complex. When N-sig disassociates, now this 2-protein complex translocates from the ER membrane to the Golgi. But that doesn't give me a transcription factor of DNA, right? But once it's inside of the Golgi, then it can be acted on by proteases that occur in the Golgi. So there are 2 proteases that are required. One protease cleaves the transcription factor from our inhibitor, right? So now it's basically released. So that site 1 protease, S1P is site 1 protease. Then we have to cleave just the transcription factor part so that it's no longer embedded and anchored in the membrane. So we have a second cleavage event by site 2 protease. Site 2 protease releases that transcription factor, and now it can translocate to the nucleus, and it can go and affect our LDL receptor. It can go and affect HMG-CoA reductase and HMG-CoA synthase. So transcriptional control is really, really important for cholesterol metabolism, right? So you have one transcription factor that's going to go in and regulate lots of different genes. Okay. So now that we kind of have a big picture, can we go back to statins? Y'all ready? Yeah? Okay. I don't want that. Okay, so let's talk about statins. We know the whole point of statins is to reduce what? Like I take this so that I don't have heart attack, right? So like cardiovascular disease. I want to take a statin and I want to reduce cardiovascular cardio disease. Whether that's just an atherosclerotic plaque or a heart attack or whatever it is, right? So what do the drugs actually bind to? To HMG-CoA reductase as what? An inhibitor. So what activity do we block? De novo cholesterol synthesis. Okay. So what happens to the intracellular cholesterol level in, let's say, the liver cell? Does it go up, down, stay the same? We would have a decrease in intracellular cholesterol, 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 and we'll say liver, right? Okay, now that we know how synthesis of cholesterol is regulated by transcription factors, a low level of cholesterol intracellularly would lead to what? So activation of SREBP precursor, right? Which is in the, it goes from the ER to the Golgi, Golgi, Golgi. Then it gets two cleavage events. And then you have a transcription factor, an active transcription factor that promotes expression of what? LDL receptor. What else? And synthase, right? You also have HMG, CoA reductase and synthase. But does this matter? We blocked it, right? We have the drug around, so we're blocking it. So this doesn't happen, right? Okay. So the only one that's really causing an effect is this LDL receptor. So what happens as we increase LDL receptor? So if we have high levels of LDL receptor on the outside of cells, what happens? Bring in LDL molecules, right? And so that will in turn lower our serum, serum LDL. Okay, here's the big one. This is the name of the game. If we can decrease serum LDL, how is that going to prevent? Sorry. Is that too high? Good. Sometimes I'm just right. Okay, so if we can decrease our serum LDL levels, because the LDL is not in the serum, right? What are we going to prevent? It's going to stop. Let's kind of go through a little bit more. We won't have LDL deposition, right? Because remember, the LDL will deposit kind of between the membranes and the arteries. Do you remember that? We were talking about that. So then what happens usually? Usually you have LDL deposition. Then what happens? You remember the immune cells come in and try to break it down? They're called the foam cells. Foam cells. They go in and they try to eat it, but they can't really eat it. And so they burst open and it's all there, right? So you prevent the foam cells, and then all these ruptured foam cells and all these deposited LDL molecules lead to accumulation of plaques, atherosclerotic plaques. Athero, athero, SCLORITI, atherosclerotic. That is so not right. Close enough. Y'all know what I mean. Y'all know what I mean. So now, if you don't have plaques, then what are you not going to have? No, usually what happens is you have a rupture of the plaque, right? The plaque gets really big and it inhibits the blood flow, hits that large thing that's sticking into the lumen of the blood vessel, right? And so it keeps getting hit and keeps getting hit, and eventually it breaks off, right? So you prevent plaque rupture. There's no plaque rupture, right? All of these are, you're down regulating all of this stuff from happening. So if you don't have a rupture, you're going to have no clot, right? And if you don't have a clot, you're not going to have a heart attack. Or what is it if it's in your brain instead of your heart? Stroke. Yeah. So that's really how statin drugs work. Statin question?
Yes, all right.
Okay.
So
Hit me.
why do statin drugs have like diminishing effects like with dosage wise? Why do like doctors prescribe like multiple statin drugs instead of just increasing the dosage? Because I know like if you increase the dosage, you get diminishing returns on them.
That's very true. That's very true. So you try to start off at the lowest dosage possible.
Right.
So you have kind of you're going to start your body's going to start upregulating the genes that are required to de novo synthesize cholesterol, right? So you're gonna take a certain amount of drug, right? And for however much of those enzymes like HMG-CoA reductase that you make, it's going to neutralize the HMG-CoA reductase, right?
Right.
So then your body's going to go, well, I'm not making cholesterol like I should be. So it's going to increase transcriptional control of HMG-CoA reductase. So now I'm going to make more HMG-CoA reductase than I have of the inhibitor, right?
Right.
So then my doctor's going to increase my inhibitor, and what's my body going to do?
And make more. Right.
So,
but why does adding another drug on top of?
Because usually it's a different chemical structure. It might interact differently, so that usually eventually you're going to get that same kind of effect. Yeah. Yeah. And sometimes they'll put you on more than one.
Yeah. That's what I was asking, like, because like if one doesn't do enough, they'll put you on another one, and it like they would have you on 3 or 4 different statins at the same time.
Yes. So some of them don't target HMG-CoA reductase. Most of them do, but sometimes they'll put you on one that's HMG-CoA reductase and one that's another place in the pathway, so that if you target 2 places in the pathway, you really shut it down. Yeah. But our bodies are tricky like that. They try to overcome whatever we try to medicate them with. Cool. Okay, so I gave y'all questions, right? Or anybody need one? Section 1 questions, remember section 1, 2, and 3? Yeah, you're like, I'll take one of those. 2, 3. Anybody else? Okay, my last one. That's okay. But you can take a picture of your iPad too, and then do it. Everybody. Oh wait, that's the wrong one. That's the wrong one. You got it's that one. Yeah, that one. You want to take the picture first? Did I give y'all the right one? No. Let me see if I have it on. Here, that's not correct. This is correct. Oh, I'm sorry. Oh, I need to put some motivation. Hold on, let me see if I have another one. Because I gave them out like at the beginning of the chapter. Yeah, it's on Moodle too. Oh, you got it? Access it on Moodle. You got it on Moodle? Yeah. All right, so we only did like the first 2 questions, right? We only did the first section. And I'll pull it up on Moodle. I can pull it up here. Here. So what I want us to do is I want us to finish the next 2 sections, and then I have another little activity, because I don't think that's going to take us the whole time. But I have another. Well, you know. All right, but Y'all can get in groups, y'all can work together. I don't want y'all to have to sit and listen to me all day today too. Yeah. Structures. Okay, so if you finish with that one, I have a few more. I'll give y'all these extra. Okay. In case y'all finish before somebody else, you know what I mean? And these are posted in the middle, these new ones are posted in the middle too. So I want us to stop and like go over answers together before we go over the second set of questions. So try 2 sections. I'm sorry, you need another question. This is the second. You have the first one? Yeah. I try to always put it on the middle. Did we go? We don't. Did we go? Yeah.
Well, I think I didn't do it, but I just did the first 2. Going over those slides.
Okay, so try to do the first sheet and we'll go over all these. I don't know where else we'll do that.
That's just.
And I'll try to pull it up here, but like it's not letting me. I might have to pull it up on the computer. I am. I keep clicking continue and it keeps cycling. So I'm gonna try and switch to the computer and do it off of here. So let me stop share. Yeah, that's for later. If we get there. If we get there, we'll see. And the next time I see you, what do we have? Yes. Okay, biochem.
Describe the physiological physiological benefits. Ketogenesis is useful for starvation.
Maybe it's a Moodle thing. Oh, there it goes. Okay. I want to download it. We'll go over them. I don't know. We'll go over them.
I didn't think we did. We just kind of got the paper and. I guess I.
Ooh, oh, that's the wrong thing. I want it in.
Yeah, I think we did do number 1, but we just didn't do number 2.
I
don't think they're all looking at.
Wait, what?
Because we're looking at section 1, question 2,
Uh-huh.
and it says mirror state, but when I look up mirror state, I see purple mirror state, unless I have to look at the book, but,
You might need to look in the book.
okay.
I think, let me see. Yeah, let me see. Let me see what page.
But it goes away with everything. What was the last one? The next party. Was it like CC?
Yeah.
Found it.
All right.
It's on page 731 in the first edition.
Okay. Let me look in the second edition. Yeah, it's gonna be. It's gonna be on a different page, isn't it? Page 765. Look, I'm trying to write with the wrong pen. Page 765. This is for second edition. What is it?
We're like, no, man, we're already here. Like, our next action is for like 2 hours.
Look up how many cars
All right, come on. Let's stick to the question.
Mirror state has 14 carbon.
Because the NAD is 2.5 times.
It's
Yeah, the answers are there.
And then half by one for action. So it's 7 Sterol, 6 and then we have to. Yeah, so I'm gonna pull it up.
Oh, use the chart.
Is it still? And then 2 NADH is
1.5.
Because every 2 is like a whole.
So I just kind of did my calculations based on page 16.1.
Ooh. Hey, Mr. Melvin. How are you? No, it's okay. Well, she's having diarrhea right now too. Yeah. So, right. So I was calling because we need to do heartworm, but like yesterday, she was, I don't know what's going on. So I don't know if you had anything today, this afternoon. I'm free starting 11:30. Like I can have her, I can be to my house and back. Okay. Yes. Thank you so much, Melvin. Okay. Alrighty. Bye.
This is like 2 carbons. That's gonna make us see a blue. So we have 14 divided by 2. And then every carbon is each increased by every 2. Okay, increased by 2 carbons. And it gives 7 acetylcholine. Let me see, but 1 less. I like chapter 16. That's why.
Oh, we did. We really did one.
Yeah.
We did one.
We did the first one of section 4.
Yeah.
And I don't remember.
We
but
did
I don't
the
have them
total
written down
yield.
as well.
And
Yeah, we didn't do the 2. We didn't do 2.
The red stripes are gonna be as much as mine.
Don't use Wikipedia.
I know. We're trying to, it's really hard first to find,
All you have to know is how many carbons are in the chain. You're doing number 2, right?
Yeah, but then it says, what's the net yield of ATP? That's what we're on. So we're looking for that in the book or on the internet. We're trying to figure out.
So figure out first NADH, FADH2, and acetyl-CoA.
That's what we're
Okay,
looking
so
for.
you
We're
have
looking
that.
For acetyl-CoA.
Okay, so then all you need is the ratio of what you're getting from NADH, FADH2, and
Yeah, that's what we're
acetyl-CoA.
looking for.
But that's in the book.
That's what I'm saying.
Okay.
We're looking for that, but I'm looking in the book, she's looking on the internet. So we'll see first, and then we can check with each other.
Okay.
the book is a very broad.
It's true. It's true.
The book gives a lot.
Yeah.
This, but you should kind of know where some things are in there.
I know it's in chapter 16.
Sort of, yeah. Yeah.
That's really only 6.
Try table 16.1.
16.1. Oh, yeah. Yeah, when I said mine was working at like 10% of what it normally does, I wasn't kidding.
Found it, the oxidation of the acetyl-CoA cycle generates 24 NADH, 8 FADH, 2 and H. So multiply that by 7. 1 FADH. Oh, wait. That is 8 acetyl-CoA does that. So divided by 8, then multiply by 7. So 24 divided by 8, 7 is 24. I don't think there's
Okay, it's like a C28.
I have a question right now, also, why one. So 7 is 21 NADH.
I'm trying to do a chart.
And 7 so that gives you. No, it's 8 times 3 or 8. So 10.5 times 5. Seven. That's 2 is 3.5 times 3 is 10.5. And then 7 more. So you have to think about it in terms of NADH, and then what is NADH?
What you got?
So, doing a common case study company
Oh.
for me.
Yeah.
Find that like as soon as we get back from spring break. Whenever you guys like.
Well, like within that first week after.
And
Yeah,
it
that's
that.
shows you.
Yep.
Is that gonna be done in class? Like. or
No.
It's just you.
Just
Okay,
me.
I was-
Because like
That's what I was
we got
like.
things to cover.
Okay.
Yeah.
Yeah.
And just answer the questions like normal and everything.
Yeah, answer the questions normal and then just present.
Okay.
Yeah, so like we just talk about when you wanna do it.
I could do it earlier than that. It's more convenient.
Doesn't matter to me.
Okay. Yeah, no, if that's, I can do it like as soon as we get back from break. It won't take me long.
Okay, so then we just need to pick a day and a time
Okay.
where it works with your schedule and my schedule.
Okay, that's what I'll do.
So sit down and look at some possibilities.
Okay.
It sends me an email and then I can look at my calendar
And
and we'll find what's works.
thank you.
Yeah.
It should be more of the 7 plus 9 was. We looked at the wrong thing. You told us table 16.1 and we went figure. So we found it, but it took the long way around. Yeah.
That's okay.
Is about 94. So it says 35. This is true. Wrong formula, right answer. Well, it was the right formula. It was just a very long formula. All right. 35 plus 9 plus 15. Section 2. Ready?
So everybody got number 2 on section 1? No? Okay, keep going.
Can we mention the page of table 16.1 on section 2 as well?
I don't know.
Section 1, question 2.
Section 1, question 2,
On the first edition, it's 783.
table 16.1. So second edition is page 819. First edition.
783.
We're trying to help people out. And it's 8. We're nice here. Okay, so.
You're supposed to show this and it's not adding up to that.
I mean, we did the calculations together, so I'm like,
It doesn't. But ours, 21 plus 7 plus 7 is on.
so. Now to section 2. Number 3. What metabolics and hormonal
No,
signals
I didn't-
control fluxation of fatty acids?
Oh, that's, yeah, so 21 times.
It's a
It's in here.
It's an interesting day today.
Yeah.
What?
So it's an interesting day today.
So I'm just glad that I don't have work because one of the students there is a hardcore teacher.
Oh, Lord.
And will like
Hands to yourselves, right?
yeah, right. My mom was like, I got my clinic tomorrow and I don't have anything for you. And I said, you're an assistant principal, just tell them if they pinch you, give them insulin. Like, duh. That's what I would have done. Okay, whatever. I know.
Yeah, but we did. So allosteric is. Okay.
So, okay. Metabolic.
I took that deliberation.
Citrate and maltol, but mostly citrate. Because I don't know. I don't remember maltol, so.
Citrate's gonna go on.
Is palmitoyl CoA just palmitate or are those two different?
No, palmitoyl CoA has the CoA attached to it.
But it's attached.
It's a
to
fatty
palmitate.
acid it's the fatty acid with the acetyl group attached to it.
So citrate. Is metabolic. 7 times 1.5. And it says palmitoyl CoA. Palmitoyl CoA is the word. Oh, palmitoyl CoA makes it inactive. So, because when you have palmitoyl CoA, that means that it's low and they can't clean the CoA with it before oxidation. So citrate and then you add 7 plus 9. Oh, fatty acids. So. Yeah, citrate means it has a high energy charge in the cell, which means it's going to start with fatty acids. Okay, acetylcholine carboxyl. I activate acetylcholine.
phosphates. All of mine. Metabolic is we always say ours are the metabolic is citrate because that is the substrate in I wish that boss would actually just look at my schedule and see that I'm in class right now. Stop texting.
Okay, don't look at your phone. Don't look at your phone.
Well, it's for my timesheet, so if I don't do this, I get fired. So I kind of have to. It's been turned in. I hope. What do they want from me? That's not enough.
There was one back in the day, I was have that.
Yeah.
And why do we always say epinephrine don't talk about cortisol? Because it's saying how cortisol hormones.
We gotta draw the line somewhere.
So we just talk, but like it could be more.
Yeah. There's so more in everything.
Cortisol when you need a fight or flight.
You have both epinephrine and you have cortisol.
So it's not just that.
It's not just epinephrine. Oh my God, there's so much more that happens.
Like epinephrine kind of embodies like fight or flight.
You can't do it all. It's the only thing. You just can't do it all.
I wanted to learn about every single hormone in the body for several years.
Well, and you could take specific classes for that.
Well, that's what I'm saying, like it'd take like several years of learning. Oh, I want to take it. Okay, I'm done with horrific stuff. Or metabolic. So hormonal is insulin, which is citrate, and that sounds so great, but it has an O. That's really hard to know. If I was here for my stomach, I would. No. So activates.
Okay, y'all wanna, let's at least talk about a few of them before we go. I don't wanna, it's 10 o'clock, so I wanna be able to make sure that we leave answering some of these. Okay, what's the total yield of NADH, FADH2, and acetyl-CoA in the complete oxidation of stearate? Assuming all of these metabolites are oxidized by citrate and oxidative phosphorylation, what is the net yield? So you had to go and look up this fat, right? You went and looked it up. So I'm gonna come down here. And how many carbons is this? It's a C,
14 carbons.
it's a C14 fatty acid, right? C14 fatty acid. So if we have a C14 fatty acid, what are we gonna generate in oxidative, in beta oxidation? I wanna say oxidative phosphorylation. You're gonna make 7 acetyl-CoA,
6 NADH.
but you're only gonna go through 6 turns to make it happen. So you're gonna make 6 NADH and 6 FADH2. Okay, so let's deal with the NADH and the FADH first. So how many ATP do I make from my 6 NADH? Well, you have to know your conversion, right? So you get 2.5 ATP, which I would give you on a test. Well, that's ATP per NADH. You should memorize, but I would give you on a test, right? Times FADH2 is times 1.5 ATP per FADH2. Okay, so how many ATP do we get from the NADH? 15. Okay, how many do we get from FADH2? 9 ATP. Okay, so now we have to take this acetyl-CoA. What happens with acetyl-CoA? It goes into the citrate cycle. So for every 1 acetyl-CoA, what do we get? You get 1 GTP, which converts straight to 1 ATP. So we can like 1 FADH2
and
3 NADH, but we're going to multiply these. I want to multiply these times what? 7. So I'm going to make 7 ATP. 1 FADH2 is going to make 7 FADH2s, but I'm going to multiply that by 1.5. Then 7 times 3 times 2.5. So how many ATP do I get from the FADH2? 10.5. And how many do I get from the NADH?
52.5.
Is that really 52? Yeah.
52.5.
ATP. Okay. Can I extend out another page? It's not gonna let me do that? Great. Okay. So what do you get?
94.
94. What's minus 2?
The activation.
Priming.
That's what I read.
So you would go like minus 2 ATP. Don't forget about that energy requirement for priming.
So it is 92. Or is it 90?
So this all totals to what? If you put this in your calculator, you get 94. So 94 minus 2, you get 92. A net of 92 net. Did it ask you for net? Did it say net?
Yeah, it's a net yield.
Yeah. Yeah, net. I was like, did I not write net? Okay. Yeah. Unless I ask you to show your work.
Double check that.
You could double check it that way.
16 more away.
Yeah, like you know what your answer should come out to.
Is that after minus 2?
You could use it to check your work. Absolutely. Absolutely. Okay. All right. So that was question 2. Question 3, I'm gonna have to like erase this stuff because it won't let me put another page because I'm in a different thing. Where's the eraser? There we go. I hate this.
I was gonna say that's
I hate
right.
this. Ah. Come on. All right, so what are we doing in this one?
What metabolic and hormonal signals control flux through the fatty acids?
So flux through
Fatty acids.
fatty acid synthesis. Okay, what enzyme are we talking about? If we're gonna regulate flux through, we're gonna talk about acetyl-CoA carboxylase. Acetyl-CoA carboxylase. Okay. Y-A-S-E, D-O-X-Y. Okay. All right, so acetyl-CoA carboxylase, what's gonna happen? What kind of regulation do we have of acetyl-CoA?
Yeah, allosteric and
There are two. One's allosteric,
nearest available.
second is,
Metabolic versus hormonal.
call this metabolic. Call this one hormonal. Okay. So let's talk allosteric first. So we're not going to modify anything. We're just going to bind stuff, right? So what binds to acetyl-CoA carboxylase? One of them is citrate.
I'm trying to not just like blurt out everything because I know.
What else? So palmitoyl, IT, what is it?
OYL.
ITOYL. Palmitoyl, CoA, right? Okay, so why are these two going to be allosteric regulators? Like you could list them, sure, but why do we want these two to be allosteric regulators? If you have lots of citrate, what does that tell you? Or-
Settled it out into the citrate shuttle and
So are we, so this allosteric metabolic control, are we saying that when citrate is bound, is it a positive or is it a negative effect on acetyl-CoA carboxylase activity? Positive. Okay. How about palmitoyl-CoA? All right, so how do these different ones work? What does palmitoyl-CoA do? The polymerization site. SITE. So remember, acetyl-CoA carboxylase is active when polymerized and either phosphorylated or dephosphorylated. Which one? And dephosphorylated.
It's probably because it's not a kinase. Kinase are usually activated by the phosphorylated. It's not, so.
Yeah, I don't have a way for that. Okay, what about citrate? Does citrate actively block the polymerization site? No, it binds at a distant site and induces a conformational change, right? So it's a distant allosteric bind. Okay, covalent. If it's hormonal, I want to know what hormones are doing what. So, okay, so insulin, start with insulin. What happens if you have insulin?
Activates protein phosphatase.
So if you have insulin, you're going to activate a phosphatase. A phosphatase does what? Dephosphorylates. I'm just going to do that as minus P, because it's easier to write. And so if you take off the phosphate group, if it is dephosphorylated, it is active. and a polymer, right? Okay, how about my other one? Glucagon, epinephrine. Okay. We're going to activate a phosphatase again? No, a kinase. So if we activate a kinase, we're going to phosphorylate, and that phosphorylation depolymerizes, and depolymerization results in an inactive enzyme. That's pretty good. Yeah? Okay. So now I need to, y'all, can I erase yet or not yet? Not yet. Don't erase. Please don't erase. All right, so, no, write it down. Hunter, write it down. You have a pencil, you have a paper, write it down. Okay, y'all ready? That's okay. We get to what we get to. Because the answers are in your book. I mean, you know, like it's not. It's just nice to talk about it.
Yeah.
It's really nice to talk about it. Walk through it. Okay. All right. So now we're on number 4. Okay. So AMPK. So this is a kinase that phosphorylates acetyl-CoA carboxylase, right? And we just said that if we phosphorylate it, what happens? Depolymerization, inactivation, right? Okay, so AMPK is a kinase. And so if it's a kinase, then you should be able to regulate that kinase activity. And it is regulated. It is a kinase that is regulated by phosphorylation. Okay, so why is it, what's the metabolic logic of having that kinase activated, right? AMP mediated activation of the kinase that will phosphorylate AMPK, right? To control fatty acid synthesis. Okay, it gets a little confusing. Okay, so you have low energy charge. That's exactly right. So if you have a low energy charge, that tells you have lots of AMP. If we have lots of AMP, what becomes active? AMP kinase is active. That's true. And instead of AMPK, I put it AMPK kinase. I don't know. But why is it active? Because what? Well, that's true. We need to make energy, but like, why is AMPK active? Like, there is a covalent change on the AMPK that makes it active. So if AMP is bound, it is an active enzyme, but it's also controlled by phosphorylation, right? So LKB1. What is that? It's a kinase, and it phosphorylates what? The alpha. AMPK. PK. So we have an enzyme that will phosphorylate AMPK. If AMPK is phosphorylated, what do we know about AMPK? It's active. Yes, AMPK. It is the kinase that phosphorylates the AMP kinase.
So when.
You remember the phosphorylation cascade?
Yeah.
It's the
So
same thing.
when AMP kinase is active,
It-
then
Phosphorylates.
it phosphorylates the acetyl-CoA carboxylase, which stops synthesis and allows degradation.
So that's a great question. So in this case, if we have a low energy charge, what do we do with acetyl-CoA?
We have to,
Do we want to go make fats?
no, we want to break down fats.
We don't want to go and make fats. So any acetyl-CoA that we have is going to be used for what?
Creating.
We want to, not for creating glucose.
Oops, trading ATP
Citrate cycle to what?
to NH.
ETC, right, to ATP. We want to use acetyl-CoA to make ATP if we have a low energy charge. So we want to inhibit fatty acid synthesis and use that acetyl-CoA in the citrate cycle. Yeah? Ask.
Is it always that? I guess, is there a pattern for like a carboxylase if it's dephosphorylated or active versus a kind? It's just each enzyme different.
Each enzyme's different. And a lot of times, if you look at the alternating homologous pathway, if you phosphorylate in this pathway, it increases activity. And if you phosphorylate in the pathway going the opposite direction, it decreases. So that's the pattern. It's not individual. You have to know this pathway is phosphorylation active, this pathway is phosphorylation inactive.
And know which enzymes go.
Yeah. Yeah. All right. So we got 2 minutes. We're not gonna tackle another one in 2 minutes. But
Do we want to finish the rest of that question?
which question?
The same, number 4, because it also
Didn't we?
talks about explain the metabolic logic of insulin.
Oh, okay. Insulin. Okay. So if we have insulin, so that means we have very high energy. Okay. Then what? Do we want AMPK to be active or inactive?
Inactive.
So we want AMPK to be inactive. Do I have any more room? Oh, good. AMPK to be inactive. We want it to be inhibited. So are we going to phosphorylate it or dephosphorylate it? Dephosphorylate it. So I'm going to put a minus P.
Because
So-
insulin stimulates the phosphatase.
Yes. So if AMPK is inactive, what does that tell us about acetyl-CoA carboxylase?
It's active.
Acetyl-CoA carboxylase. Acetyl-CoA carboxylase is phosphorylated or dephosphorylated?
Dephosphorylated.
Dephosphorylated. If it is dephosphorylated, then it is active acetyl-CoA carboxylase. And we're going to use that and we're going to go and make what? Sorry, y'all. Forgot about the second half of the question.
Speed brand.
Good. Try some of the other ones.
Number 5, we already did a class together.
Did we do 5 in class together?
Yeah, how to statin drugs reduce serum LDL levels and risk.
Oh, yeah. Okay, so we did that. We did 5. So we only missed 1.
Yeah.
And we really talked about,
The files thing,
Yeah.
yeah.
are you file records?
So we
Resins, yeah.
we have sort of talked about all of them. Not so many on the second set of questions,
Oh, no.
but that's okay. What do we got next time? I see you.